Today I shall discuss about the structure, seed structure and its germination in chapter 6 of icsc syllabus class 9 standard, and in the section of unit 3 plant physiology. Then in this class I shall discuss about the what is seed, what is fruit, what is grain, and what is the different type of seeds, and structure and function of monocot seed and dicot seeds and their differences, and as well as the germination of the seeds, how many types of germinations and their difference. Today we shall discuss about the process by which a seed is formed within ovary. This is the seed. The seed is the main structure by which a plant produces a small seedling or plantlet. So, how the seed is formed? The seed is formed by the process of fertilization, by the fusion of the male gamete as well as the female gamete. This is the ovary. Within the ovary, ovules are present. Within the ovule, embryo sac or female gametophyte is present. When the egg of the female gametophyte is fertilized by the male gamete that come into the egg through pollen tube, then the male gamete fertilize with the female gamete, then this process is called fertilization, and after fertilization ovule is transformed into seed. So this is the formation of seed. Then the question arises how the fruit is formed. Fruit forming process. This is the same process by which after fertilization. The ovary, after fertilization, the ovarian wall transformed into a special structure. This special structure is called epicarp. This epicarp, when the wall of the ovary transformed into epicarp and the ovule is fertilized and it became a seed, then the ovary is matured and ripened and fertilized. This ripened, fertilized ovary is called fruit. What is fruit? What is fruit? What is fruit? Fruit is the process by which a ovary is ripened after fertilization, after producing the seed. Let the question arise, what is grain? Grain is the process. 3 questions in first paragraph. The grain. What is grain? Grain. Grain is the fertilized ovary containing fertilized ovule, that is seed, but the seed coat as well as the fruit wall is fused. Grain, this is the covering of the ovary. This is the covering of the ovarian wall. Within the ovarian wall, seed is present. Seed also covered by seed coat. This is green color is seed coat, and the other color, pinkish color, is the fruit coat, the fruit wall and the seed coat when fused. In this case, the fertilized ovary is called grain. So, what is grain? Grain is the condition, it is the fertilized ovary, it is a fertilized ovary when seed coat and ovarian wall is fused. This is called grain. So grain, example of the grain, there is the grain. Example আমরা যদি দিই, example কি হবে? Rice and wheat. Rice and wheat. Here both case the ovarian wall as well as the seed coat is fused. They united. They cannot be separated from each other. This is the grain. The three questions: what is seed, what is fruit, and what is grain? Then the question is arise that what is the how many types of seed, and on the basis of their size, seed types. What are the seed types? There are several type of seeds on the basis of their size. Large seed, very large seed, at a very large size, very large seed. The size is very high. Medium seed, and the third is the small seed. Very large seed is the coconut. Coconut is a monocotyledonous plant, and this fruit is fibrous, and within the fruit the stony covering is present. The seed is very large and covered by a stone like wall. Then the medium size seed, the medium sized seed is the mango seed. It is large also, mango seed, or it is a custard apple seed, medium size seed. That another name is the phoenix sylvestris, is the data palm, the medium sized seed. And very small size seed, this small size, a very teeny seed, just found in the fucus. This is another name is the bot, fucus. Sometimes some seeds of the orchid. These seeds are very small. Amorphophilus monocot seed is very small in size. On the basis of their size, large seed, medium sized seed, and the very small side, minute seed. The minute seed mainly found in the orchid family, some ficus family. These are the size. Then on the basis of their endosperm, seeds are divided into two types. Endosperm, on the basis of the endosperm, how many types of seeds? How many types of seeds on the basis of endosperm? On the basis of the endosperm, seeds are divided: endospermic, non-endospermic, albuminous, non-albuminous. The first is the albuminous seed, and non-albuminous What do you mean by endosperm? Endosperm is the tissue which contain huge amount of food materials that provide during the growth of the embryo. Endosperm, in the previous class it was discussed that how endosperm is formed. The endosperm, this is, suppose this is the embryo sac. Within the embryo sac, the central cells are present. Opposite the three cells, the egg cell and two synergids, three antipodal cells, and central cell contain two polar nuclei. These two polar nuclei when fuse, forming a definite nucleus. Then when the male gamete enter into it, one male gamete fuse with the egg, forming the zygote, and other male gamete enter into it and fuse with the definitive nucleus, forming a endosperm tissue. This endosperm mother cell is formed. From the endosperm mother cell, the general divide and redivide, forming a endosperm. This endosperm is containing numerous food material. This food material is supplied to the growing embryo for its development. So what is the endosperm? Albuminous means endosperm containing seed, and the non-albuminous, the endosperm absent. The question example, albuminous seed, that is the endosperm containing seed. Most of the monocot seed is albuminous. Monocot seed. Most of the monocot seed is albuminous. The wheat, rice, these are the endospermic monocot seed. And some few dicot seed are endospermic. That is the castor. The dicot, this is a dicot, and these are monocot. Castor is a seed that contain endosperm. Endosperm containing seed, castor. Monocot seed is gia maize. This is also a monocot, also is albuminous, gia maize. Gia maize, wheat, rice, castor, all are endospermic seed. First three are monocot and castor is the dicot. Some other dicot seeds also contain, sometimes may contain the albumin endosperm, that is the poppy. Poppy seed is also contain endosperm. Another endospermic dicot seed is the custard apple. Annona squamosa. These also contain albuminous seed, although this is a dicot plant. Most of the dicot plant is ex-albuminous but non-albuminous. Ex-albuminous or non-albuminous seed, generally most of them are dicot. Mango, guava, mango, and other dicot plant, all are generally ex-albuminous. But few dicot plants also having the endosperm, that is the custard apple having a endosperm. Which dicot plant do not have endosperm? Dicot plant, most of the dicot plant do not have endosperm. Which dicot plant contain endosperm? Custard apple or castor having endosperm, they are dicot plant. But few dicot plant generally, that is that in this case the albuminous plant and non-albuminous plants are divided on the basis of their endosperm. What is the function of the endosperm? Endosperm provide the nutrition function of the endosperm. What is the function? What is the function of endosperm? It provide nutrition to the developing embryo. This is the question. Then the type of some structure of the seed. Some structure of the seed. Monocot seed, dicot seed. Two type of seeds are generally found. One is the single cotyledon and two double cotyledon. What is the structure? At first we shall discuss about the materials which are present in the seed. Seed, this is a monocot seed, maize seed. It is actually grain. It contains outer fruit coat and inner seed coat. This is a grain, where ovarian wall fused with the seed coat. This seed is a grain, monocot grain. But another seed is the bean seed. This is a kidney shaped seed, and this seed generally found in the leguminous plant. The bean is the legume plant. What is the bean seed structure of the bean seed? Bean seed is a seed having two coverings. Outer covering, seed covering, is called testa. Outer seed coat covering is called testa, and the inner coat covering is called tegmen. Testa and tegmen. It has two surfaces. One is convex surface and other is the concave surface. That is why it looks like a bean shaped structure. It looks like a kidney shaped structure. Outer testa, inner tegmen. This testa is very hard and it protects the inner part of the seed. And the innermost part is also it is thin, but it also have the protective function. In this testa and tegmen, a elongated structures, a triangular remnant scar, white scar is present. This scar is called hilum. What is hilum? Hilum is a scar by which a seed is attached within the ovary by funicle. This is a scar. So suppose we consider a ovary. This is the ovary. The leguminous fruit is called pod. Suppose we draw a pod. This is a pod. In the pod, fruit of legume plant, in the plot, this tissue is called placenta. This is a placenta area, soft tissue. soft tissue that's found in the one margin, and from the margin several small stalk develop, and each stalk attached with a seed. This attachment site, when the seed is matured, this seed is separated from the stalk and leaving a scar. This is the scar, and this scar is the hilum. In the above side of the hilum, a small aperture is observed. This is a small aperture is observed. This aperture is called micropyle. This aperture is called micropyle. So what is micropyle? Micropyle is the aperture through which in early stage male gamete enter into the ovule. In later stage it present as a remnant part. During germination, the external water and air enter into the seed and helps the germinate, helps the growth of the embryo. So this is the seed, outer seed testa, inner tegmen, and a scar is present when we view in this side. When we view the front, this is the concave side, and in the concave side a scar is present. This is a scar, and at this scar a aperture micropyle is observed when we view in this way. This is the dicot seed, external bean seed, external structure. Then we consider the internal structure of a dicot seed. Later we shall see the internal structure of the monocot seed. What is the dicot seed? Dicot seed, the main characteristic feature of the dicot seed, it contains two cotyledons. So seed, the covering of the seed is testa, and the inner covering of the seed is tegmen, and it contains two cotyledons. These are the two cotyledons. These two cotyledons are attached with each other by the axis. These axes have two parts. This is the red color, this is the cotyledon. One cotyledon, two cotyledon, and this is the embryonal axis. Two cotyledon and embryonal axis constitute a whole embryo in dicot. The upper part, this upper part, and this is the lower part. The upper part is called plumule. This upper part is called plumule. And the lower part is called radicle. Total structure is the embryo to one cotyledon. This is the cotyledon 1, cotyledon 2, and this is the axis. Upper part is the plumule, lower part is the radicle. And the upper zone, upper part of the embryonal axis just adjacent the plumule is called epicotyle. Epicotyle. And lower part of the axis is called hypocotyle. Epicotyle and hypocotyle. Epicotyle is the adjacent part of the plumule, and hypocotyle adjacent part of the radicle. What happens when the embryo grow? The plumule that gives the first leaf, This is the grow shoot, form SHOOT, shoot, stem, and this grow and ultimately into the root, transform into root. This is the main structure of the dicot, and total cotyledon, when it is folded, it remains in folded condition, and then it's covered by inner tegmen and outer inner tegmen and outer testa. Outer testa and inner tegmen. This is the main structure of the dicot bean seed. Then we discuss about the structure of monocot seed. From the term monocot, the cotyledon number is 1. This seed is the maize seed, M-A-I-Z-E, maize seed. What is the structure of the maize seed? Suppose this is a seed. Most of the seed, it is a seed coat, and outer the fruit coat, as it is called grain. The most of the seed is filled with reserve material, living tissue for storage food. This is called endosperm. In dicot seed, endosperm is totally absent. 2 cotyledon is present, and here the most of the space is occupied by endosperm, and a epithelial tissue separate this endosperm from the embryo that is present at the lower side. What is the embryo? Embryo is, this is the embryo. This is the embryonal axis, and this is the lower side, and a single part is present. This is called cotyledon. This is single cotyledon, and the upper part is called plumule. This is called plumule, and this is called radicle. Plumule and radicle both are covered by 2 special structures, protective structures. This is called coleoptile, and lower part also covered by another covering. This covering is called coleorhiza. Coleoptile and coleorhiza, this is called the embryonal axis, and this part is the single cotyledon. This cotyledon is called scutellum. So this is a special structure. This a small elongated structure also found. This is called the epiblast. So this is what is the difference between monocot and dicot. Monocot seed contain, the question arises, difference between monocot, difference between monocot and dicot seed. First difference is the having 2 cotyledon, having single cotyledon. This cotyledon is called scutellum. Second difference is the having endosperm in monocot, dicot have no endosperm. Third is the embryo is short in size, it is rudimentary in size, but here the embryonal structure is very large, total size is embryo. The epicotyle the coleoptile and coleorhiza. Epicotyle, the plumule is covered by coleoptile and radicle is covered by coleorhiza. But here no such special structures are observed in dicot. A albuminous cell layer, epithelial tissue layer is present here. Here it is not present. Here the sometime chia seed, our seed coat and the fruit wall is fused. Here the fruit wall is totally different. The total seed, several seeds are present within a special fruit called pod. The pod is the fruit. Pod is the fruit of the bean. And this is the seed of the bean. A pod containing 5 to 10 or 12 seeds within a single pod. But here this is a single grain and several grains are arranged in a female inflorescence called cob. So this is the main two difference between monocot and dicot. Then the question arises, what are the factors that are responsible for the germination of the dicot and monocot seeds? Several factors. At first, to understand about the factors for germination of seed, what is the germination? What do you mean by germination and what do you mean by dormancy? Two terms is observed after preparing a seed within a fruit. When fruit is ripened, seed is matured and fall into the ground, it do not germinate, it do not grow and form into a small seedling. Why? The process is called dormancy. Dormancy. What do you mean by dormancy? Dormancy is the living status of a growing embryo within seeds. But in this case, embryo do not transform into small seedling or do not grow into a plantlet. Do not seed, but do not transform into small plant or seedling. This is the dormancy. Several chemicals are present in the seed. These chemicals prevent the seed for its bursting of the seed coat and come out the growing embryo. This dormancy, when after some time period the embryo, suddenly the seed coat of the embryo is ruptured. Some enzymatic activity also develop within the seed. At the time when the seed get sufficient amount of oxygen, temperature, water, and in some cases embryo, then the germination will occur. After what are the different steps? Formation of the, what are the different steps during a plant growth? First is the development of the ovary. Development of ovary. Then the development of ovule. Along with the development of ovule. Then seed, fruit and seed develop by fertilization. Seed gets maturity and enter into the dormant period. Later from the dormancy, germination takes place. These are the steps for the developing a small plantlet. Later stage, why the all factors, oxygens, temperature, sunlight as well as the water, why all these are required for the germination of the seed? Before that, how a seed germinate. Suppose this is a seed. This is a seed. This is a sisal seed. This seed contains a hilum and a micropyle is present. Seed is covered by seed coat. Seed is covered by seed coat, and when within the seed, the cotyledon, within the seed, cotyledon and embryo are present. This is the seed. When the oxygen and water enter through this micropylar aperture, then water when enter into the embryo within the seed, then the different enzyme formation occur. Different enzymes due to the cellular activity increases. This cellular activity when increases, huge amount of water intake is performed, and when the water is sufficiently rich and then some cellular substances are prepared, then the cotyledon gets larger in size and create an external pressure. When water enter into the seed and oxygen, it get oxygen, cellular activity increase. Within the cell of the cotyledon, several substances are formed, some substances and huge amount of water intake occur, and the size of the cotyledon gradually increase. This create a external pressure. Due to this external pressure, the outer seed coat is ruptured, and the latter it is degraded, and the total seed cotyledon gradually increase its size, and the radicle grow and plumule grow upside. This is called germination. Then the question, which factors are responsible for germination? This is called the temperature. Second is the oxygen. Third is the water. And fourth is the PGR. Some PGRs are also responsible for seed germination. And when the seed germinate, on the basis of the germination type, plants are differentiated into two categories. Which category? The germination are two types. How many types of germination? The question arises, how many types of germination? How many types of germination? Germination are two types. First is the Epigeal germination, epigeal germination, epigeal. Second is the hypogeal. Epigeal and hypogeal. What is the epigeal and hypogeal germination? What are the epigeal germination and what are the hypogeal germination? Epigeal germination and hypogeal germination. Epigeal germination generally a germination where the cotyledon enter into the air. That is, it looks like it is found in the bean. Epigeal. Epigeal germination, suppose a seed, bean seed, when a radicle grow and plumule grows. In later stage, this side is elongated more faster. As a result, the two cotyledon ultimately it enter into the air by the growth of the epicotyle. By the growth of the epicotyle, the seed cotyledon enter into the air. What happened in this case? This is the root. This is the root and the cotyledon. First, the cotyledon enter into the outside and then the plumule grow. The plumule grow and the first leaf and the stem develop by and pulling the first the cotyledon of the seed in the air. This is called the epicotyle, epigeal germination. Epigeal germination generally found in the bean plant. It is also found in the tamarindas, tetul plant. And hypogeal germination. Hypogeal germination means the total seed cotyledon remained buried in the water, but the radicle and plumule both grows equally in both direction. Radicle enter into the soil, plumule enter into the air. Radicle form the root and the plumule form the shoot. What happened in the, this is called the epigeal and later is the hypogeal. What happened in hypogeal germination? In hypogeal germination, suppose this is a gia maize grain. It germinate. At first it produce a plumule and later it a radicle. In later stage, the seed also remains under the soil, but its plumule grow. Plumules grow and enter into the atmosphere, and the radicle grow, enter into the soil. This is the leaf. And it grow, and in this case the root develop. So here the cotyledon remains attached with the growing plumule and radicle and remain buried under the soil. This is the hypogeal germination. This is called hypogeal germination. This is also found in the Caesar aritinum. This is the chola. This type of germination. Later, the question germination and later the another type of germination, epigeal and hypogeal in general type of germination, and other type of special type of germination generally found in mangrove plant. What is the mangrove plant? Mangrove plant is a halophyte. They grow in the marshy land, particularly in Sundarban, where water is saline. Salinity of the water is very high in the salt water. The Sundarban, what is the mangrove forest? Mangrove forest is the main forest that protect the sea bank from the erosion and protect our North Chobbish Pargona district from the tide. These plants have a special type of two features. First feature, some plants having prop root and some of the plants having the respiratory root. The root by which it can get oxygen from the air. This is the exceptional root that do not enter into the soil because of the high salinity of the soil as well as the low oxygen presence. Due to the low oxygen and high salinity, the root grow upward, forming a respiratory root. Suppose this is a clay land and marshy land, and all are contains water and huge amount of salinity is present. The plant grow, but their root do not enter into the soil due to the salinity and lack of oxygen, and these root grow upward. This is called the respiratory root. But here the root is not a question. These are the adventitious root. But the question is the germination. Because of the salinity of the soil, the plant, we are the dicot plant. When the dicot plant get a fruit, within the fruit, the seed is germinated in the tree. It do not detach from the tree without germination. When it detach, it must be germinated. During this case, what happened? This is a seed. This is the plume. The radicle grow in the lower side and like a curve-like structure. The lower head is very large and extended in diameter, large diameter, and the base part is narrow. And the upper part is small, grow the plumule and a very small minute stem arises. In this stage, it is shed, it is separated from the plant and it fall into the marshy soil. And in the marshy soil, the lower plumule, cup-shaped plumule embedded in the soft soil, and the latter plumule it remains embedded in this condition, and when it grow, then the stem grow upward. This type of germination is called the viviparous germination. Viviparous germination. Just like viviparous animal. What is the viviparous animal? The animal which give birth of a young baby from its womb. And why they give birth? Because they prevent their embryo from their external harsh environment. The mammal is this group of animal where they give birth of small baby, just like this type of mangrove plant. Which is the name of the plant? Soneraria. Soneraria. Rhizophora. Soneraria, Rhizophora are different mangrove plants. In this case, the seed is germinated when it is remain attached in the fruit. Because if the seed is fallen in the marshy saline soil, it do not get sufficient oxygen and cannot be germinated. That is why the special type of adaptation of the mangrove forest, in this case the seed is germinated in the air by getting the